All right. Good morning, everybody. I'm going to take you on a little journey that we had a couple of years ago that basically AstraZeneca decided that we had to get into the genomics game. And how were we going to do that? And we thought, well, we're going to need a bold ambition to put forward because there's a lot of other bold ambitions already out there. So, there we are. Good. Okay. So we decided, what the heck? We're going to announce that we are going to collect 2 million sequences on patients and subjects from across the world. And part of that was going to be 500,000 samples from our own clinical trial samples that we are going to start sequencing, which we have already sequenced tens of thousands. But then joining like the UK Biobank efforts, we'll have access to another 500,000 there, Genomics England and others. So we are building a consortium of various partners, both academic, government, as well as industry partners, to put together a cohort of at least 2 million sequences over the next 7 to 8 years. So you can imagine this is quite an endeavor. And we had to create a special or a new organization to run all of this data. So it's called the Center for Genomics Research at AstraZeneca. And they are really focused on collapsing analysis. So one method to look at, can we associate rare variants with disease and basically try to find new targets. Okay? But what we're doing is forgetting a lot of other questions that people in our organization want to be able to answer. Okay? So yes, we want to use this for discovery. We want to find new targets. We want to find new ways of looking at diseases because many of the diseases we are trying to fight are not a single disease. Many of us remember the days where cancer was cancer and then all of a sudden it became lung cancer and brain cancer. But then in the late 90s, early noughties, technologies such as gene expression actually showed us that now we can sub-phenotype these diseases down into very specific molecular phenotypes. Therefore, understanding what are possibly the new targets that we should go after to address those sub-phenotypes of disease. So we really want to learn more about the biology. We also want to make sure that we have good development with our drugs. So how are we actually getting value out of our drugs and getting that value to the patients? Which patients are going to respond to our drugs? Which patients are not going to respond? And those that do not respond, what can we understand about those molecular mechanisms to now come up with new drugs? And of course, the last one is launch, making sure that we are launching drugs and getting them to the right patients at the right time. That's all great. And that's what the company basically does for a living. But at the same time, we have to start thinking about other things. Could we predict adverse events based on genomic patterns? Could we stratify patients for clinical trials, getting those patients in the clinical trial that actually will respond to our drugs, or at least we think will respond to our drugs, based on these molecular profiles that we're developing? And so the CGR basically had no predisposition to say we're going to share the data with the rest of the organization, that we are going to make these genomics data, along with the associated clinical data, environmental data, anything that we can get with actually our therapeutic scientists. And I saw that as a big problem. And so when we look at the broader picture, we want to make sure that our scientists have the ability to use these data to the best of their ability. And what do they want to ask? They want to ask questions like this. I'm not going to go through every single line that's up here. There's no reason to reread it. But the fact of the matter is our scientists want to do more with that data than just try to find a new target or to validate that target, such as neoepitopes or neoantigens that we heard about yesterday. What they want to be able to do is they want to understand mode of actions. They want to understand how the immune system, especially at my company, is actually changing with respect to the therapeutics that we are now offering. So there's much broader questions that we're trying to answer around this. And how do we get our head around providing tools that are easy to use, have a great user interface, get people excited about the data that they're seeing, and want to go in and do more and more to answer these questions? So this really comes down to population genomics. And one of the things when we were looking at how are we going to do population genomics, we started talking to several companies in the space that claimed they had interfaces that would allow us to do some of this. And to be honest, there are companies out there that do. But when we looked at those companies and we looked at the software and we looked at the clunkiness of those tools, we came to the conclusion that only bioinformaticists could ever use it. And the trouble is bioinformaticists won't use it because what are they going to want to use? They're going to want to use what you just saw in the last demonstration. They're going to want to use the Jupyter Notebook. They're going to want to bring Dockers in so that they can run the analytics that they want to run without having to go through the push-button GUI interfaces. Bioinformaticists hate that. But at the same time, population genetics or population genomics only works if all of the scientists that have expertise in this area are able to look at it and bring their own judgment into the data analytics that we want to do. So it's what genes are associated with our disease of interest. What diseases are associated with my genes? How are you going to actually look at that? Which genes are associated with response and non-response to drugs? And then the non-responders, like I said, getting down to the molecular mechanisms of what's going on. What are the clinical parameters associated with the GOI? Okay? So really, this has to do a lot with how are we going to get this out to our researchers. And honestly, I didn't have an answer because everything that we were looking at just was not fit for purpose. And of course, then we also, how do we get things into clinical trials? Again, not going to go into all of this, but how do we actually make clinical trials more effective so that they're cheaper, they're quicker to run, they're more targeted to the patient population? So this is when I started meeting up with Brady Davis. So for those of you who do not know Brady, he's the Chief Strategy Officer for DNANexus. He and I knew each other for years, back when he was working for Oracle and I was working for Moffitt Cancer Center. And we had become friends, and he came to me saying DNANexus was thinking about a new direction for the company to go. And it was really around how do we take population genomics and start using it at scale and getting it out to the researchers, especially the bench scientists, who are the experts in their therapeutic areas. So we talked about this for a while, and we decided, let's put together a project plan, see if we can't come up with some ideas, and actually make this thing a reality. Because we really want to do exactly what I said before as this trajectory. But it really comes down to is making sure you have the security, the compliance in the data, everything that you need, that if you use any of this data to go to an FDA or an NHCE submission, that you have all of that data ready for you and you're able to get it to those agencies very easily. Because our long-term vision is really putting the power or empowering our scientists to do this work. A bioinformaticist runs around the company, and we do a lot of work for a lot of different people. So one day we're oncology, the next day we're respiratory, the next day we're cardiovascular. We don't get into the depth of the knowledge of the diseases that our therapeutic area scientists do. And so we want to make sure that we have a virtual cohort visualizer. One where our researchers can quickly choose all of the parameters that they would be interested in from the phenotypic side, whether it be a diagnosis, whether it be a pathology report, whatever that might be. And then look at the samples genomically that match that criteria, and then start being able to look at the variants. Or go back the opposite way. How can we look at a certain variants in genes that we find very interesting? Ooh, it's in an upstream UTR that may have something to do with the regulation of that particular gene. Is it having any effect? So not only looking at just the genomics piece, but eventually being able to look at the transcriptomics and even the proteomics piece to understand more of the system rather than just the individual modalities. This gives us the best guess at what is happening as far as the pathway and the genetic networks that are occurring, and hopefully that will lead to better understanding of disease and eventually getting the right drug to the right patient at the right time. So it sounds simple, right? Sounds like, oh yeah, just do this and life will be good. Not so much. I mean, we had to get this really into a very detailed project plan where it was the MedImmune scientists, especially from my team, working with DNANexus scientists, and hashing out the requirements over months and months and months till we came to an agreement where we could start actually making this tool a reality. And in fact, you just saw pieces and parts of it in the demonstration right before here. Okay, so this is where we started. We basically started by saying we needed a cohort explorer, okay? And we worked out various details of what we needed in there. One, we insisted that we needed analytics. Number two, that it had to be a really good front-end user interface, something that people are going to want to use and not get bored with. But we also knew there were some trade-offs, because just as you saw with the GWAS analysis that was just performed, couldn't do it in real time, and that's always the problem with live demos. Even if things can be done in real time, network connections at these kind of places just fall apart. But they had a good backup. So, we basically decided user interface, analytics had to be embedded in there. We did not actually talk about Jupyter initially, but we're very happy that that was something some of their other partners were interested in doing. And in fact, my bioinformatics scientists are very excited about having that now. We had to have it so that we could quickly put together GVCF files so that we could do a population VCF and have that available for exploration of the phenotype or vice versa. And so we basically went through this whole program and entered it into a co-development project. And basically, DNANexus did all of the programming, where my team really helped with the requirements, getting everything we needed in the system for phase 1, and then, of course, we'll be expanding that into the phase 2 in the future. And so you might say, okay, well, where are we? Well, we're right at the point now we're going to be doing end-user testing to basically look, or end-user acceptance testing, looking at the product as it exists today, providing feedback on any bugs and anything we find. And what we're hoping is by the time the UK Biobank data starts to roll out in December of this year for the partners that have agreed to go into that partnership, that we will be able to now use the DNANexus TIP for our scientists to be able to explore that data. And so this is kind of the beginning of the project. Come on, got to point it in the right direction. So again, we define goals, features. We've done several development iterations. So the nice thing is the DNANexus scientists come out to Gaithersburg. We work with them on not only how we develop the requirements, but looking at the tool and giving them advice on the tool. And I will say this has been an extremely nice partnership to get us where we have been so far. We started this in January with a lot of earnest. And it really has only taken us about 9 months to get a product out the door. And I really have to say kudos to DNANexus for putting the time, the effort, and making sure they brought on the resources to actually get us to this point. And I'm very excited to roll this out to my scientists at MedImmune, and soon they'll be able to have access and empowerment to this genomics data that we are collecting. So what are the major deliverables? You've seen some of this already. We have had the data models and ingestion of data. So it's not just about how do you get genomics data in there, but it's also how do you get the phenotypic data in there? Because as you understand, any of you that work in this area, that if you've seen one EMR, you have seen one EMR. I know it's an old joke, but it still holds very true. That the way that individual hospitals or academic medical centers have set up their electronic medical records is not standardized. Even if you're all on Epic or you're all on Cerner, the data and the way it is handled is very different. So there has to be very flexible ways of ingesting the data along with very flexible data models to be able to map to that data model from your EMR or from your other sources of phenotypic data. Like I said, we needed a cohort browser, an analysis framework, and we'll continue to build on the analytics framework that was already shown to you, a visualization framework in UI, so being able to visualize the results like in Manhattan plots or volcano plots or other things like that, and being able to manipulate those plots in real time. And so that was all a requirement. And we wanted an out-of-the-box analysis app for the first phase, something that our researchers could just click a button and get an association, and that is what you saw with the GWAS study itself. The platform has to be scalable. So one of the things that we talked about early on is we're looking at 2 million sequences over the next 8 years. I don't want to build something today and 3 years from now being looked for another product because this doesn't scale. We discussed it. Of course, they have to scale for the fact that they're going to have the UK Biobank data and 500,000 samples in the very near future. And so we felt that this was something feasible, basically, especially based on the architecture that they put forth in the cloud. We had to have sharing and collaboration functionality, just like you saw Doctor and Geek up here earlier. You have to be able to share results you see and do that in a collaborative manner and very seamlessly. If you have to constantly go into different tools, like Fan was saying earlier, researchers get bored. They don't want to have to do that. They want to stay in one environment just like a physician does in the EMR system. We had to have good admin, security, and compliance. I mean, we're dealing with a lot of different countries in our project. So this is not just about the UK, Sweden, and the United States. This is about patients from all over the world, from all of our clinical trials. And depending on where that data was generated, we have very, very different rules on how that data can be used and accessed. And so we had to have a system that allowed for all of those different compliance regulations to be incorporated into the tool itself. And data federation is another huge part. So one of the things I'm very interested in, and I've been very interested in for a long time, is something called leaving data in place. As data continues to get bigger and bigger, we have to find ways to leave the data where it is and bring the analytics to the data. But when you're talking about consortium where there are some partners that will not let you take the data outside of their environment, Genomics England being one, of course they're rethinking that now, but still, that you have to go into their environment. What you want to invoke an analysis once, not multiple times in different environments. So how do we now create a federated analytics environment that allows us to do the same analytics across multiple sites without having to do it individually each and every time? The other thing is, many times clinical data can't be brought out of a company's environment, such as AstraZeneca, where we want the clinical data to live inside our firewall. Again, having federated querying capabilities to be able to bring that data from an existing source behind our firewall into the analytics tip so that we can do the analyses we want to do, but it's certainly just a transient federation, meaning the data goes away after you stop using it. Now, we're not there yet with this particular functionality, but this is functionality we will be building during phase 2. Has to be interactive analysis. Data federation part 2 will bring it up to the next level. And then, of course, we'll have additional data analytics tools out of the box that will basically be a one-click once you choose your cohorts that you want to analyze. And so this is just an accelerated development process just to show you where we started, where we are today, and where we plan to go in the future. And we are very excited to bring this tool, like I said, to our researchers to empower them to use these data. What we've accomplished so far, we've worked together on the requirements, data workflows, and APIs. So that was very fruitful. We agreed upon the scope of phase 1, which was not easy, but I think we came to a really good place. Even though there were adjustments made during the project, the roadmap stayed consistent as far as what we were trying to get to. We basically chose the user interfaces were being designed, so we helped with those, as well as collaborated to define the analytical methods. We are now discussing phase 2 plans. Like I said, we're almost ready to get into user acceptance testing. I mean, it's only a week and a half away, and we're going to be doing a lot of testing for the next couple of months. And then we plan to deploy late this year in 2018, latest in Q1 2019. Like I said, why are we doing this? What our researchers want to get to is the fact that we want to stop using model organisms as much as possible. And you might say, well, but that's what pharmaceutical companies do. That's true if you're a small molecule company. If you're a biologics company, it's not as true. And the reason is because many of the times the drugs that we are developing, which are antibodies or peptides, but especially antibodies, basically do not work in rodents. So if we try rat models or mouse models, the immune systems between that and the human are so different that many times we have to actually create surrogate antibodies that are more mouse-like or more rat-like so they actually work, but it's not the true drug that we then be giving to the human. And so what we want to do is start moving more and more towards the human model organism. As new organoids come out and other technologies come out to be able to look at human tissue, human cells in real time and in real microenvironments, it is getting more and more exciting about doing this type of work. So doing systems biology approaches, genomics, proteomics, metabolomics, and so on these types of systems is going to be important. But then there has to be a way to compare it to the human condition itself. And that's where the AZ genomics efforts come in. By having 2 million sequences, we hope to be able to take the human-like experimentation that's going on and then translate that directly into the human with the various omics modalities. And what we're hoping is that DNANexus will be that main platform for the analytics back to the bench scientist and to the bioinformaticist. I know we've heard this before, but this is a main focus of the company right now, is taking our data and making it fair. And what does fair mean? we had it yesterday, but I'll go through it quick. Findable. Your researchers can find it. They can access it if they actually have the permissions to do that. It is interoperable, meaning that you can use that data across multiple systems to answer different questions. And it's reusable. Yes, you can answer different questions with the same data. Now, this takes a lot of metadata, a lot of semantics, ontologies, controlled vocabularies to make data fair. But this is something that we feel is necessary, not just for the type of work we're doing today, where it's looking at genomics or looking at transcriptomics in silos. What we're hoping is that what this will actually create is a way for scientists to reuse data across therapeutic areas, doing an analytics across multiple clinical trials or multiple research experiments at one time, so that now we will have the ability to use machine learning and deep learning technologies very readily. Remember, garbage in, garbage out. Fair data in, hopefully good results out. At least that's the plan. And this fits into our larger strategy of basically bringing all of this together. So, yes, we have all of these different modalities that we are collecting data on. We do have a data integration strategy called integrative informatics that we're bringing this all together using the semantic web and linked data technologies. But again, we want to make sure that not only do our researchers have the ability to look individually across all of these silos of data, but then actually be able to bring this data all together to be able to analyze it as one whole cohort, looking at the systems biology approach. We believe that this will get us to, again, the right patient, the right treatment, the right time. So you think this might take an army to do. I don't have the DNANexus team listed here, but great interactions, great collaborations with them. I think you will see the fruit of our success in the very near future and being able to use these tools for various things. But I need to really thank Dominic, Chat-Chien, Andre, Bev, Matthew, Rachel, Tim, Wen, and Xiao Tao for their great work over the past year. And again, we had a great time, and I think now that we are able to empower our scientists, the genomics data we are collecting will now be able to be utilized and will actually show some real value. So thank you very much, and I'm happy to take any questions.